LVV. I'll talk about in vivo CAR T introduction and challenges in tLV design, and also challenges in process development. And also we'll mix up this part with our ProBio solutions. And then we'll talk about regulatory requirements for in vivo CAR T LVV, which is tLV, targeting LVV. And then I will talk about the basic information about ProBio and the sites we are where we provide LVV services. So in vivo CAR T is actually making CAR T cells directly inside the bodies of the patients. So we don't have to isolate the T cells and then transduce it with a vector to let it have a CAR gene. We directly give the CAR gene to the patients. So the gene goes into the T cells directly in the patient's body and make the CAR T cells in situ. That's what we call in vivo CAR T. In vivo CAR T, normally we use two types of vectors to deliver the CAR genes directly into the patient's T cells. The most commonly used ones are lentiviral vectors and LNP. Lentiviral vector has some advantages. The most important one is the most important one is that lentivirus can integrate its gene into the genome, so the therapeutic effect will be very long term. Sometimes it can last for 10 years, several years, as long as the transgenes is not silenced by the patient's genome. But there is also risks for lentiviral vectors. Sometimes it gets integrated into the oncogene, the loci of oncogenes, and will activate the oncogene expression. So, but we will try to limit this risk. So here are some pipelines that are already available in the market, and we have to we can focus on the two pipelines highlighted by the yellow. So these tools are the ones that have already disclosed their phase 1 trial data. So one is called Clonia, and another one is ESO-Biotech, which is already owned by AstraZeneca. There are some challenges in making LVV vectors for making CAR Ts. First of all, there is challenges in designing the tLV vector, and also which includes the envelope designing, transfer plasmid designing, and CAR molecule designing itself. And also there are challenges in the upstream process of LVV, which is actually packaging the LVVs in 293D cells. And also there is challenges in the downstream process of LVV, which is actually purifying the LVVs. So I'll talk about these challenges and the solutions of ProBio. So how does lentivirus infect cells? Our conventional lentivirus we use is pseudotyped by VSVG. The envelope protein is VSVG. So VSVG is doing two jobs on the lentivirus. So the one job is it will recognize the receptor of itself, which is the LDLR. LDLR is present on a wide range of cells. So that's why LVV pseudotype with VSVG can infect a very wide range of host cells. So after the VSVG bind to the LDLR, and the virus will be swallowed by the host cells, which is called endocytosis. And after the swallowing, the virus stay inside the endosome. And after the pH in the endosome decrease, the VSVG will change its conformation and cause the membrane fusion between the LVV envelope and the endosome. So the capsid of the LVV will be finally released into the cytoplasm of the host cells. This is the whole process of LVV infection. So as we can see here, the VSVG is actually doing two jobs. One is recognizing the receptor on the host cell. Another job is to mediate the endosomal escaping, which is actually the infection of the virus. This is how VSVG is mediating the infection. So the first picture here on the left shows that VSVG trimers can bind LDLR on the host cells. So this is when the pH is still normal, which is neutral pH. So after the cells so after the virus is bound to the LDLR, it gets swallowed by the cells into the endosome. The pH will decrease because there are proton channels on the endosomes will increase, which will decrease the pH. After the pH decrease, as we can see, each monomer of the VSVG will be unfolded, and then this arm here will be inserted into the endosomal membrane, and then the VSVG will refold itself, and then physically pulls the endosomal membrane closer to the viral envelope. When they are close enough, the membrane fusion will take place. The most important thing we have to remember here is LDLR, which is the receptor of VSVG, shows up on very wide range of cells. So if we use VSVG as the envelope protein of LVV, the LVV will infect almost all kinds of cells in our body. So that's why when we try to deliver LVV, the CAR gene, into T cells directly, specifically in the patient's body, we have to let the LVV go find the T cells by itself. We have to make a specific LVV, which can target T cells specifically. To do that, we have to maintain the membrane fusing function of VSVG. We should let it still mediate the endosomal escape, membrane fusion between the envelope and the endosomes. But we have to remove its receptor binding ability. We have to detarget it to let it be blind to its cognate receptor, LDLR. So we do some mutations. For example, K47 R354 mutations on the VSVG, so it won't be binding its receptor anymore, but still it can mediate the fusion. But who is doing the receptor binding job? Then we will let antibody or scFv or VHH do that job. We just put a transmembrane domain onto scFv or VHH, then it can be anchored on the envelope of LVV. So the scFv or VHH, which are called T cell binders, can bind the T cells. So the binding job is now done by the anti-CD3 scFv, for example here, T cell binder, and after the endocytosis in the endosomals, VSVG, a mutated VSVG, is still doing its membrane fusing job. So now we split the receptor recognizing and membrane fusing into two molecules. One is doing only one job. Each is only doing one job. So when a client comes to us, normally they will have the design of the envelope. Normally, they will have a binder, T-cell binder, sometimes targeting CD3, sometimes targeting CD7. If it's targeting CD7, then the LV will be binding T cells and natural killer cells as well. And also, they will design a fusel gene. Normally, we'll still use the VSVG because this one is the most validated one. It's pretty safe in the clinical trials. And we just mutated to let it be blind to its receptor. And also, clients normally will have a CAR molecule design. To design a CAR molecule, normally we will consider which kind of promoters we will be using. Normally, we use a human constitutive promoter, which is strong, such as EF1 alpha. And also, if we want to add another safety layer onto the vector to let the CAR gene only be expressed in T cells, we can use T cell specific promoters to express the CAR gene. And also, we will have to have a signal peptide in front of the CAR molecule, for example, to direct the CAR molecule onto the membrane, onto the T cell membrane. Also, transmembrane and intracellular co-stimulatory domains will also be considered when we design a CAR molecule. Here are some examples from the top benchmark players of the in vivo CAR T companies. The first one is the design from Umoja. We have to mention Umoja is because Umoja is one of the pioneer companies, pioneer players in the in vivo CAR T field. Even though its phase 1 trial isn't that smooth, we have to still remember that they are the first ones who introduced this conception to CGT field. And the second one is the second one on the right is the design from ESO Biotech, which is owned by AstraZeneca now. They're using an anti-TCR VHH as a T cell binder because TCR is specific on T cells. So it's a nanobody which can recognize and bind TCR. And also they still use VSVG mutation as the fusel gene to mediate the membrane fusing. Also, they overexpressed CD47 on 293 cells, so the CD47 can be inherited into the LVV membrane. CD47 can signal to macrophage that about don't eat me. So the macrophage won't be swallowing a lot of LVVs after we give the LVVs to the patients. This will increase the efficacy of LVVs when we actually use them. And also they remove the MHC1 complex from the 293 cells. So the LVV produced from these cells will not have MHC1 complex either. This will decrease in the immunogenicity of the LVVs produced from these kind of 293 cells. Also, regarding the CAR itself, they use a T cell specific promoter to drive the CAR expression instead of constitutive ones such as EF1 alpha. This will make this CAR gene only be expressed in T cells. There are some risks to use tLVVs in the patients. The most important one is that if we try to package a tLVV in the 293 cells, the GOI is normally co-expressed during the packaging process. The protein will show up. The GOI protein will show up in the 293 cells. Normally, the protein is a transmembrane protein. For example, here are CAR, certainly a CAR anti-CD19 CAR. So because a CAR molecule also has a transmembrane domain, it will be anchored onto the membrane of the 293 cells, and then during the budding of the virus, this anti-CD19 CAR will be inherited onto the tLVV membrane as well. So the CAR also has a CD19 SFV on it. So it can also perform as a binder, but only this time it binds to CD19. So this tLVV, we wanted to have a CD. Sorry. We wanted to only have CD3 binder to bind the T cells, but now, because the GUI is already expressed into a protein, it can bind CD19 now as well. So if we inject this kind of LVVs into the patient's body, it will bind not only T cells, but also B cells, which is actually the cancer cells, and let the cancer cells also express a CAR, which will make a CAR B cell. This will mask the CD19 antigen in the cells and let the cells escape the cell therapy. And the example here on the left bottom corner here shows that only a single B cell which escaped the CAR therapy will lead to a cancer relapse later. So if we want to remove all the entire population of B cells, we have to make sure the CAR molecule doesn't go into the B cells. And to do this, our ProBio designed a lentivirus which can repress the CAR a GUI expression, the protein production during the packaging. So a molecule such as CD19 CAR will not show up during the packaging process. And this is actually what we call silent in production, but active in therapy. The CAR molecule only expressed after the LV infect the patient's T cells. As we can see here, during the packaging of LVVs in 293T, we can barely detect the expression of CAR protein, but after we have the LVV and infect the T cells, JERCAS cells, we can see the expression of these CAR genes here. So this will make the LVV envelope cleaner and safer for using in vivo. And also there are some difficulties about packaging the tLVVs. For example, if we use insufficient binder, this tLV will not be able to bind T cells. But if we use too much of the plasmids, which will express the binder, which will let the binder be too enriched on the envelope of LVVs. And then there are some spatial hindrance between the binder and the fuser gene, and this will also cause the difficulty of the LVVs infecting the host cells. So a very optimal amount of binder is very important to make efficient tLVVs. To do that, we have to perform DOE experiments to make sure the plasmid ratio is optimal. Okay, so here is an example of a DOE assay. So the one here shows that different factors here: transfer plasmid amount, Gagapol plasmid, Rev plasmid, fuser gene plasmid, and binder plasmid. And we adjust these accordingly and see the titer, the P24 production of LVVs, and choose the optimal one. And there is also, in the past, I was talking about the upstream challenges, and there is also there are also challenges for downstream process. The first one is because we now have binders on the LVVs, this will make the tLVVs more fragile for all kinds of purification processes. And also there is, because we're using them in vivo, now we have a terminal sterilization filtration. And during this process, we will also lose some production. And the last one is impurity. This is because the vesicles produced by 293T cells, along with LVVs, normally have the same size with tLVVs. So it's kind of difficult to completely remove them. All these are the challenges for downstream process. But if we do some optimizations on these factors, for example, we adjust the workflow, we control the time period for purification, we do some shear force management, we do some ionic strength optimization, or formulation optimization, we can increase the recovery of LVVs. After we do this optimization, we can increase the fax titer normally 3 times in our process. So there are challenges in all aspects of LVV production, upstream production, upstream challenges and downstream challenges. But we will work with the clients to go through all these optimizations to make sure the recovery is awesome, or at least acceptable. For the regulatory requirements for tLVV, the most important thing we have to remember is that moving to tLVV, the lentivirus are used as drug products, not a drug substance anymore, not a medium of drug production. It will be used in a final form. So there are some specifications specifically for drug productions if we use tLVVs as a drug production. So there is visible particles, particulates, insoluble particulate, deliverable volume, insoluble particulate, and particle size. There are also other specifications here, insoluble particulates and endotoxin, and these are specifications we have to consider on a case-by-case basis. And these are the detailed standards for these specifications. These are mostly the standards from US Pharmacopeia. And another important thing is if we use tLVVs as drug production instead of drug substance, there is no definition of drug substance anymore during the production of LVV. In other words, we don't have to define a drug substance when we try to produce tLVVs for in vivo CAR T. At least FDA has this kind of definition. In the morning, Jennifer already gave a very good introduction about ProBio, CDMO service. We have 3 sites global-wide. We have one in Nanjing. Nanjing is mostly for antibody discovery and antibody production. And if you're ordering LVV services from us, we will be providing the services mostly from Zhenjiang site. And hopefully in near future, we can also produce LVVs in the US, in Hopewell, New Jersey. So we can provide, which is called one-stop service for tLVV CDMO. You give us the design, molecule design, and we will produce the plasmids. We'll produce the tLVVs, and we do the CMC manufacturing, quality controlling with you, and provide you GMP level products. And in ProBio, we can do some packaging test. If you just enter this field, you can come with us with some candidate molecules. We can do small scale packaging test to see which one is giving you pretty good titer tLVVs, and also we can provide DOE experiments to try to get the most high titer ones from the plasmids you provided us. So, as I said before, if you're buying tLVVs from us, we will produce them in Zhenjiang site right now. So in a PD lab, process development lab, we will provide the scales of 2 liter to 54 liter for adherent 293 cells. But if we use the suspended 293 cells to produce tLVs, we provide 2 liter scale and 50 liter scale. And for clinical GIP and the GIP manufacturing, we will provide a 10 liter scale and 200 liter scale for using suspension system. So this is pretty much the information I wanted to provide the audience. So if there's any questions, I will be happy to answer.